Hi and hello everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s Bioneet. By the last video, we successfully completed the central dogma of molecular biology, which includes the three major events. Those are replication, transcription and translation. We have discussed these three events in detail in both prokaryotes and eukaryotes in the previous videos. Today, in this part of videos of molecular basis of inheritance, we are going to start a new concept, that is a mechanism of gene regulation. A small request from my side, kindly watch all my videos made on this molecular basis of inheritance for better understanding of the concepts. And one more request is, please like and share the videos and subscribe my channel. Let's get into our today's topic, the mechanism of gene regulation. Generally, a gene, by undergoing a transcription and translation, involves in the expression of a character in the organism. We know very well about this in central dogma of molecular biology, where a segment of a DNA or gene involving in the expression of a character in the organism, first it undergoes transcription, right? By undergoing transcription, it synthesizes an mRNA molecule. This mRNA molecule being synthesized then undergoes translation in order to produce a linear polypeptide chain of sequence of a specific amino acids according to the sequence of triplet codons present on the mRNA molecule. This polypeptide chain being produced here is actually inactive. In order to become an active enzyme, this linear polypeptide chain undergoes post-translational modifications. These post-translational modifications forms an enzyme of tertiary or quaternary structure like this. This is what we call an active enzyme. This active enzyme involves in a particular biochemical reaction. We know very well that each biochemical reaction is catalyzed by a particular enzyme. This enzyme, whenever involves in a biochemical reaction, it results into expression of a character in the organism. What is happening here? A character in the organism is being expressed whenever the gene undergoes a transcription and translation, which means the gene expression includes the two events of central dogma of molecular biology, transcription and translation, followed by expression of a character in the organism. Here the question is, do all the genes in DNA of an organism expresses their character always? The answer is no. Why? Because the organism, a body of an organism, whenever it requires the expression of a particular character, then only the gene responsible for that particular character involves in gene expression. If body of the organism doesn't require the expression of a character in the organism, then the gene of particular character won't undergo transcription and translation. It remains suppressed. This is what we call suppression. Expression of a character is called gene expression, or non-expression of a character in the organism is called gene suppression. What is that mechanism that is controlling, that is regulating the expression and suppression of a gene in the organism? Actually, that mechanism is the gene regulation mechanism, which was explained by the two scientists. Those are Jacob and Monod. These were the two scientists who first explained the mechanism of gene regulation by taking the lac operon. They explained what? Lac operon for the explanation of a gene regulation mechanism in E. coli cells. E. coli is a prokaryote. This lac operon gene regulation mechanism is a model gene regulation mechanism in all prokaryotes. Today in this video we are going to discuss this gene regulation under three headings. Those are, one is key terms, the scientific terms in order to understand the lac operon and mechanism of lac operon. And the second heading is structure of lac operon in prokaryotes. The third heading is mechanism of gene regulation in lac operon. First, let us start with the key terms related to the gene regulation, so that we can understand the structure of lac operon and mechanism of gene regulation in lac operon. What are those terms? One is operon. What is this operon? Operon means a group of genes, in DNA of the organisms involves in regulation of a function. The group of genes involving in the regulation of a function in the organism is said to be as operon. Here we are talking about lac operon. What is lac operon? Lac operon means a group of genes involving in the lactose metabolism. This group of genes involving in lactose metabolism is said to be as lac operon. Here, based on the mode of regulation, the genes in DNA of the organism are categorized into two major groups. Those are inducible genes, and the other one is constitutive genes. First, let us discuss what are inducible genes. The inducible genes are the genes which involves in the expression of a character by undergoing transcription and translation in the presence of an inducer. Simply we can say inducible genes requires the inducer for their expression. Okay, those type of genes are called inducible genes which requires inducer. The best example for the inducible genes is a lac operon mechanism. In lac operon mechanism, what happens? The gene involving in a lactose metabolism undergoes transcription and translation. By undergoing transcription and translation, this lac operon synthesizes an enzyme beta galactosidase. The function of this beta galactosidase is to hydrolyze the lactose into glucose and galactose. Here, whenever the organism, a body of the organism is having abundant lactose, then it requires the synthesis of beta galactosidase, so that the particular gene, by undergoing transcription and translation, it expresses the character. What is that character? The synthesis of beta galactosidase. That beta galactosidase hydrolyzes this abundant lactose into glucose and galactose. If there is no traces of lactose in the E.Coli cell, then the body of the E.Coli cell doesn't require this beta galactosidase enzyme. So that the particular gene involving in the synthesis of a beta galactosidase won't undergo transcription and translation, remain suppressed. What is the inducer here? The lactose is the inducer. If lactose is present, the particular gene of beta galactosidase involves in expression. If lactose is absent, the gene of a beta galactosidase won't undergo expression, it remain undergo suppression. So what we learn here, the inducible genes are the genes which requires the inducer. The most of the genes in eukaryotes, almost 95% of the genes in eukaryotes are the inducible genes. And one more point we have to keep in our mind is the lac operon is an inducible gene regulation. Let us come to the fourth one, that is constitutive genes. What are these constitutive genes? The genes which doesn't requires inducer, which doesn't requires inducer. The best example for this constitutive genes are enzymes of respiration. Enzymes of respiration. A respiration is a continuous process. If respiration stops, the organism will die. So that this enzymes of respiration, that doesn't require any inducer, they shows a continuous expression. Such type of genes are called constitutive genes. Only 5% of the genes in eukaryotes are constitutive type. These are the key terms we have to keep in mind for understanding of the structure of lac operon and mechanism of gene regulation in lac operon. Now let us move on to the second heading of gene regulation, that is structure of lac operon. The lac operon includes the two types of genes, those are structural genes and regulatory genes. Say suppose for example this is the segment of DNA lac operon and Z, Y, A are the three structural genes. I inhibitory gene, P promoter gene and O operator gene are the three regulatory genes. Structural genes are those which by undergoing transcription and translation synthesizes polypeptide chains or enzymes. Regulatory genes are the genes which do not involve in direct synthesis of structures or polypeptides but involves in the expression or suppression of these structural genes. Hence the name regulatory genes. First let us come to the structural genes. The segment of DNA or the gene which involves in the synthesis of a polypeptide chain or a structure is called a cistron. So cistron is a unit of DNA. What is cistron? The segment of DNA or a gene that involving in the synthesis of a polypeptide chain by undergoing transcription and translation. Like cistron, there are other units of DNA. Those are racon. Racon means this is also a unit of DNA which defined as the unit of or segment of DNA that undergoes a recombination during the process of crossing over. We can also consider this racon as the unit of DNA. Another one is muton, the segment of DNA which can undergo mutations. You know very well about the gene mutations where a single nucleotide of DNA can involve in mutations. So that a single mutation also considered to be as a muton in the case of this one. Racon is the unit of DNA which undergoes a recombination. Cistron is the unit of DNA which involves in the synthesis of a structure polypeptide. Muton is also a unit of DNA which can undergo mutation. Based on the length, the racon is the largest unit of DNA. Next to this, a cistron, and a muton is the smallest unit of DNA. Leave about this. Let us come to these structural genes functions. What is the function of Z, Y, and A structural genes of lac operon? The function of Z gene is to synthesize an enzyme beta galactosidase. You know very well that the function of beta galactosidase is to hydrolyze the lactose into glucose and galactose. And the next gene, Y gene function is to synthesize an enzyme permease. Permease is the enzyme which allows the lactose into the cell so that the beta galactosidase can act upon the lactose. And the third one is A gene, which involves in the synthesis of an enzyme thiogalactoside transacetylase. The function of this thiogalactoside transacetylase is to transfer acetyl group from acetyl CoA to the lactosides, so that the beta galactosidase can recognize the lactose and act upon the lactose to hydrolyze into glucose and galactose. Not only beta galactosidase synthesis here, along with beta galactosidase, there are other two types of enzymes synthesized during this mechanism of Operon. The second type of genes are the regulatory genes. What are the regulatory genes? Inhibitory gene, promoter gene, and operator gene. This is a sequence of this. You have to remember the sequence of these regulatory genes and structural genes on Operon for NEET point of view. First, let us start with the promoter. What is promoter? The promoter is the region where an enzyme RNA polymerase can bind to the operon, so that after the binding of RNA polymerase, the initiation of transcription and translation starts, finally resulting into the synthesis of an polypeptide chains, beta galactosidase, permease, and thiogalactoside transacetylase. So promoter is the region where the RNA polymerase gets binds to the operon and initiates the transcription and translation. The second one is inhibitory gene. Inhibitory gene is otherwise called repressor gene. What we call this? Repressor gene. Because this inhibitory gene, it continuously synthesizes repressor proteins. The repressor proteins are the tetrameric proteins. Whenever these repressor proteins go and bind to the operator, the initiation of a transcription stops. Whenever these repressor proteins doesn't bind to the operator, the process of transcription and translation goes on for the synthesis of these three enzymes. Here the point is, the inhibitory gene is a type of constitutive gene because it continuously synthesizes the tetrameric repressor proteins. And another third regulatory gene is operator gene. Operator gene and promoter genes are overlapped here. Remember this. The operator gene acts as on and off switch. Whenever the repressor proteins binds to the operator, transcription process won't stops. Whenever the repressor proteins won't bind to the operator, the transcription process initiated. Hence, we call this operator gene as on and off switch. So here we learned the function of these structural genes and functions of these three regulatory genes. This is all for about today's video. In the next video, we are going to talk about the third heading of a gene regulation mechanism, that is the mechanism of gene regulation in Operon. Until that, keep watching my videos and subscribe my channel Vidya Amrutam Ashnate.